Experience is one thing you can't get for nothing. Oscar Wilde. A very good evening to one and all present here. I would once again welcome you all to the second part of our webinar. Kindly note, due to some last minute inconveniences, our second speaker, Dr. Anita Chawan, could not be with us today. However, we are very grateful to Ms. Priyanka Sigar for being here with us on such short notice. Before we begin today's talk, I would request our HOD, Dr. Hemlata Ramchandran, to say a few words.
Yeah, so welcome all, good evening, and we are very happy to have the second session. And I'm sure most of the students to whom I was interacting today, especially the TYBSc students, they are very happy to hear about what are the prospects after graduating and after the master's program. So I'm sure that today's two sessions will also be equally interesting. You will have a lot of questions to ask. And I'm happy to introduce today Jarvis, who has been in our department for a master's in life sciences, specializing in neurobiology. And we were often hearing about his work because he was associated with ICER Trivandrum. And currently we have also come to know that in the lockdown period, along with the research, he's also doing some analysis with regard to the COVID data collection. So that tells you that as a research student, sometimes you have extra clinical related work also. Whatever comes up in the lab, you have to take up those responsibilities. So we are happy to have Jarvis today with us, and I'm sure you will have plenty of questions to ask about his entire journey after completing his master's. So Asta, over to you, and then the session can continue. Thank you.
Thanks for the kind introduction.
Thank you, ma'am. Without any further delay, I would now like to take this opportunity to introduce our first speaker for the day, Mr. Jarvis Fernandez. Mr. Jarvis Fernandez completed his master's in life sciences from Sophia College. He is currently pursuing his the School of project during his master's, deciphering the effect of VPA on autophagy in vitro in Dictyostelium discoidum, the AX2 strain, from August 2013 to April 2015. He even completed an ACTRAC on a project entitled to clone shRNAs targeting profilin and cofilin in a TRFP expressing vector at Dr. Saurabh Dalal's lab in the year 2012. He worked on an honors research project with C. elegans as a model organism for a duration of 8 months at St. Xavier's College, Mumbai. He was mentioned for a project titled MIR 184 modulates neuronal excitability in Drosophila melanogaster at a research conference in January 2020. He was even awarded the Augmenting Writing Skills for Artillery Research Award for Best Story. He won the Best Presentation Award at Jigyasa in the year 2012 for a presentation of project work titled To Study the Effect of Various Factors on Development, Growth Rate, and Reproduction. He has several publications in some renowned journals. Apart from this, he has been currently volunteering part-time at the COVID testing facility at ICER Thiruvananthapuram. We are very pleased to have you with us, Mr. Jarvis Fernandez.
Thank you.
Before we move on, I would just like to request our participants to kindly read from putting up any greetings on the YouTube chat. Kindly use the chat box to type in any questions that you have, and those will be addressed after the talk. I would now request Mr. Fernandez to take over.
Yeah, Asa, can you hear me properly? Because your video was getting cut on and off. So can you all hear me properly? Yeah?
Yes, we can.
Yes, we can.
All right.
Yeah,
so
we can.
Share my screen. Okay. Can you all see?
Yes, it's visible.
Okay, so I've titled my talk So Near Yet So Far, and that's because I'm almost on my way to complete my PhD. So I hope to graduate in the next 1 year. And therefore, I'm like on my way to the home plate, which is basically the end of my PhD. Currently, I'm at Dr. Jishi Varghese' lab at ICET Trivandrum. And like every one of us, my journey first started outside my window, actually. Looking at nature, that was the spark that, started my curiosity or in and my, what should I say, adventure in science. This spark probed me into thinking about, what are the factors or that regulate various things. And later on in my life, I started actually working with fish during my younger years, so during my 10th standard and stuff. So I started keeping various kinds of fish, and the work I was looking at was the different modes of reproduction in these various different kinds of fish, with some being egg live bearers, some being egg scatterers, some actually taking care of their young. And this was like the catalyst which pushed me into research, because I was constantly wondering why, evolutionarily, why are there different strategies, in terms of reproduction. I also got interested in looking at nutrition, because nutrition is also, it has a huge role to play during development. And that's something that I actually carry forward later on in my life. So, like every one of us, one of my first official projects during my bachelor's was actually making wine. So, this is actually a picture of us in St. Xavier's College making wine as a part of a practical course. And my first official project as such was to look at, was an honors project, was to look at the effects of metal toxicity in Caenorhabditis elegans or C. elegans, and the effects of glucose and sugar-free natura on the growth, development, and reproduction of C. elegans. And this also I carried out at St. Xavier's during my bachelor's. This was where I was introduced to actually a model organism like C. elegans, which we had obtained from Sophia College. And that's when I realized that there's a college like Sophia's, which is actually doing research at the master's level. And they're looking at various aspects of uh, development, as well as neurogenesis in C. elegans, as well as in Drosophila and zebrafish. So this is something that actually I kept in mind as I was doing my bachelor's. Further on after that, I started my a research internship at Dr. Surat Dalal's lab at Actric, where what I was doing was I was basically cutting out a fragment from one particular plasmid and inserting it into another plasmid. That was all my job. That was my entire 2 months that got spent doing this. And after this, what I'm trying to explain here, which will come later on, is I've had a myriad of different experiences where I'm looking at the different projects that I've worked on. And all of these projects had different techniques that I needed to master to work on those projects. Later on, I was faced with a curveball. The curveball was my grandmother actually was diagnosed with Alzheimer's. And
I'm sorry to interrupt you.
yeah.
Actually, we can't see the presentation.
Oh, you can't see the presentation.
Yeah, it's only the title slide is visible. Yeah. Okay, now it's visible.
Okay. All right.
Yeah, that's fine.
Okay. So I'll start again. Huh? Okay. All right.
Thank you.
Yeah, just 1 minute. I just will check. Okay. Okay. I don't know if you can see. Okay. All right. Can you see it now?
Yes.
Yeah. Okay. But it's not full screen. All right. Fine. Okay. So yeah. Once again, I'm sorry for the delay. So my title slide is "So Near Yet So Far," and that's because I'm almost on the verge of completing my PhD. Like I was telling the professors before, I am actually submitting a paper today to a journal. So hopefully that will get accepted soon. And therefore, I have titled my site "So Near Yet So Far," but I'm on my way to the home plate, which is completing my PhD. I am currently in the lab of Dr. Jishi Varghese at ICET Trivandrum. And like every one of us, my journey first started outside the window, when I look outside, our journey always starts with nature. And I was always curious to know about different the wildlife that was present around me, since we lived very close to the RML colony as such in Mumbai. And this was the spark that put me down this path. And later on in my life, towards the end of my high school, towards 10th standard, I started keeping fish. And thereby I got interested into reproductive biology, because I noticed that different kinds of fish actually had different modes of reproduction. So I started with live bearers like guppies, and then I moved on to egg scatterers like zebrafish, finally bubble-less makers like betta, and cichlids, which actually take care of the young. So this was actually a very good catalyst, and actually prodded me into thinking about the evolutionary reasons for different, reproductive strategies that different organisms employ. So, like every one of us, when I joined my bachelor's, I started with my first official project, which was making wine. This is with some. This is an image with some of my friends and classmates at the time, and we are all making wine together at St. Xavier's College in Mumbai. So this was one of my first official projects as part of a BS, being in my bachelor's. Uh, my first research project was to look at the effects of metal, lithium, in Caenorhabditis elegans, and the effects of glucose and sugar-free natura on the growth, development, and reproduction of C. elegans. This was carried out at St. Xavier's College during my bachelor's. Once again, I got the C. elegans line was obtained from Sapphire College, Mumbai. And that's when I realized, this was my first experience actually working with a model organism. And that's when I came to know about Sapphire College and the research that is being carried out at Sapphire College. So this research was carried out with the help of Dr. Radhya Patra Gupta, who actually helped us publish this work in Explore, which is the Journal of St. Xavier's. So later on, I progressed into a summer internship at ACTREC at Dr. Surat Dola's lab, where basically I was actually cloning a fragment from one particular, basically cutting the fragment out from one particular plasmid and inserting it into another plasmid. The curveball that I was hit towards my TYBSc was that my grandmother was diagnosed with Alzheimer's. So, like any Indian parent would tell us, like my mother told me, Jarvis, why don't you start working on neurons and aging, and that would be something that you would honor your grandmother with. And like any Indian child, I said, okay, mother, and I rationalized it with myself, saying that okay, neurons are basically just specialized cells. So even though I was on path to work on cell biology, I changed my path and started working on neuro and decided to work on neuro. And that's when I joined Sapphire College. And I was looking at the effect of valproic acid on autophagy in Dictyostelium discoidum. So apart from the research work, I was also looking, meaning I also made a lot of friends, okay? And yeah, among my friends, here you can notice there's only one guy, there's Leander. So I think what's really important during your master's is that you need to have a good friend circle. And yeah, what I remember is like Leander and I used to travel. So he used to live at Borivali, I used to live in Malad. We used to travel in the train together. And while traveling in the train, we used to discuss a lot of science. And we also used to talk a lot about the net projects and things that we were looking at. So CSR net was something that I realized or came to know about during my master's. And seniors like Leander, Durga, and Nabila, and they pushed me towards applying for CSR. And during this application process, and they were always along to help us push us in the right direction. So what I'm trying to emphasize here is that one needs to have a close set of friends who would help push you and who would help you to actually work in the right direction. Yeah, once again, I actually missed the point. So before I actually joined the masters, I attended various interviews and various exams, entrance exams, like the TFR exams, the JNU exams. Actually cleared and passed the JNU exam. And one of the things that I would like to tell you is give these exams and then go and check the college where you're allotted. You know, if you're going through JNU, you're going to be allotted a college, and you need to go and check the college that you've been allotted. Okay, so that was something that I did, and that's when I realized that I do not want to work in that particular college. I'm not going to name the college as of now. So at Sapphires, we also went for various conferences like the IN, the Indian Academy of Neuroscience conference. These are all my lab mates at the time, classmates as well as seniors. And one of my most exciting experiences was when we actually got to visit the brain bank. And here is a picture of me in my younger days holding a actual human brain. And that was really one of the highlights of the trip, to be honest, for me, that I got to actually hold a human brain. Uh, yeah, after this, I actually moved on and I started looking for PhD positions. Like I said, I was applying for CSIR net and I had actually cleared CSIR net. So after doing that, I actually had to go around the country giving interviews at various places. So I went from ISET Pune to NBRC to CCMB and then ISET Random. And I was offered a PhD opportunity at ISET Random in Dr. Jishu Varghese' lab. So I started my PhD here in ISET Random. But what I didn't tell you about ISET Random when I joined was that it hadn't relocated or located, meaning hadn't started working, functioning in its permanent campus until then. So I actually started my PhD at the temporary campus, okay, here, listed here, the College of Engineering. And we finally moved to our permanent campus in 2018. And that's the picture of my lab. And this is the picture of the campus, actually. So the campus is located at the base of the Western Ghats. It's a really beautiful campus that we have here. So my first project was to, like I said, I was inspired to look at neuroscience, and I was very adamant that I'm going to look at lifespan and aging. So when I joined the lab, I told Jishi that I would like to work on lifespan and aging. And he said, all right, we can start a screen on the role of microRNAs that play a role in lifespan and aging. So that's what I started. And this is a picture of the brain, the Drosophila brain that I had imaged a while ago. And the image you see in green, that's actually the mushroom body, the L-shaped structure that you're seeing, that's the mushroom body, which is actually the memory center of the brain. All right. And the side lobes are the optic lobes, as you can see here. So basically, a lot of things are conserved in Drosophila and humans as well. Apart from genes that are conserved, even the sensory system and everything is also conserved. So I'd like to give a brief introduction on microRNAs because I know you all guys are all bachelor's and master's students. So microRNAs are 21 to 23 nucleotides long non-coding RNAs, which basically are complementary to the 3' UTR region of a mRNA. And if it binds to the 3' UTR of the mRNA by complementarity, it leads to its degradation via the RIS complex. So we employed a method called a microRNA sponge line, which consists of perfect microRNA complementary sites. Okay, so this basically means that this particular microRNA binds to these sponge lines much more efficiently than it binds to the mRNA target. And if it does bind to the microRNA sponge lines much more efficiently, you can titrate the microRNA away from their normal targets. And using that, we started out a screen where we were looking at the role of microRNAs in lifespan. And what we discovered was a particular microRNA X, which I'm not going to name as of now, because we are still in the process of publishing a paper, is this particular microRNA actually downregulation of this microRNA led to a decrease in lifespan specifically in the males, while as it didn't lead to any change in lifespan in the females when you downregulated in the brain. And this was something really interesting. So one thing more I want to point out is that like I was saying before, we were in a temporary campus before. And like all the problems we associate with the temporary campus, our incubators that we were keeping in our flies in broke down. And my flies actually got fried at 50 degrees Celsius and they died. And that was very heartbreaking. And that's when I realized I actually had to change my project. And so next, I'm trying to figure out another project, which was to look at the role of microRNAs in an activity rest rhythm, which is also carried out in an incubator, but in a separate incubator, which we were not using for this purpose. Now, let me explain to you what an activity rest rhythm looks like. This is what the activity rest rhythm looks like. There are two peaks of activity. There is a morning peak, which is at Zeitgeber time 0, which is when lights come on. And there's an evening peak, which is at Zeitgeber time 12, which is when lights go off. Okay, so what we found was when we decreased the levels of a particular microRNA, what we saw was that there was a decrease in the morning peak activity, and there was a decrease in the evening peak activity. This was very surprising, and it was very interesting as well. And we tried to figure out why there is this change in so these flies are not waking up, and they were also sleeping in the evening. So let me give you an example. Let's just say we travel from one time zone to the other. What happens is when you travel across from one time zone to the other, you can actually get jet lag. And the jet lag disappears over time. And how does this jet lag disappear? This jet lag disappears because there is a resetting of your circadian rhythm. And this resetting is actually done by various cues that you get from the environment. Okay, the cues that you could get would be light coming on during the day because of sunlight, and there being no light at night. There are other cues as well that the organism receives from the environment, which is for us, it's food, the food timings, because we eat breakfast, lunch, and dinner. There are food timings. There's also temperature because it's warmer during the day and it's cooler at night. So such various factors help entrain our circadian rhythm. And this input is actually received by a particular set of neurons and it's conveyed to the key circadian neurons or the circadian neurons in that organism. So in Drosophila, there's a set of circadian neurons called the morning cells, which actually receive this signal from the optic lobe. So you can see that there's a projection out towards the optic lobe. Can you see the white dots? And this is actually sending a signal to these PDF neurons, which are the morning cells here, which receive the signal. Upon receiving the signal, this PDF neuron actually sends the signal back to all these, not back, it actually sends the signal out to all these other clock neurons and helps reset their circadian clock. What it actually does, it tells them that it's morning now. And how it actually does this is actually it releases a neurotransmitter saying that, okay, it's lights are on now. This particular neuron is very peculiar. It could do this in two ways. It could release the neurotransmitter only when lights come on, okay, and not do so at night. That's one strategy, but this neuron is very peculiar. What it actually does is that it actually makes and breaks synaptic connections in the span of a day. So what it actually does is it defasciculates at night, so it makes more connections in the day. It makes more connections during the day. At night, it breaks off all the connections and actually bundles together at night. It's fasciculated. So this is how it looks like during the day at Zeitgeber time 2, which means 2 hours after lights come on. You can see it's actually defasciculated. It's actually making more connections. And at night, it actually bundles together and makes less connections. When we downregulated this microRNA, what we saw was actually in the day, it's actually making less connections. And at night too, it's making less connections. But when we overexpress this particular microRNA, what we saw was it's actually making more connections during the day and the night. Okay, so that's given over here, and we have also quantified it in the tabular format over there on the right. We next also looked at if these microRNAs could actually control neuronal firing and neuronal activity. And we looked at the GFP reporter, and what we saw was when we overexpress this microRNA, there is an increased level of expression of GFP, which indicates that actually these neurons are actually firing more. And this was actually published in the DST Avsar 2019. It's a short story wherein I explain the work to a layman. Like I was once again saying before, challenges are a part of any PhD and any research everywhere. And unfortunately, these incubators too broke down. They didn't exactly break down, but basically the CPU unit actually got spoiled. Okay, meaning in the circuit, there was a problem in the circuit. And what this led to a huge number of problems wherein we are seeing light come on at different times of the night, unregulated, which was not actually scripted. And this led to a disruption in the experiments, and I had to actually stop this work until we could repair the system. And that's when actually our lab was actually shifting from the temporary campus towards our permanent campus. And I realized I need a project wherein I don't actually rely on any external factors, like I do not require equipment like an incubator to actually function, and I can utilize simple things like a microscope to get things done. And that's when I started a third project, the role of microRNAs in growth and metabolism. And we were looking at the role of a particular microRNA, which upon down regulation led to a larger fly. So you can see that this fly actually becomes larger in size. And we have weighed it out here, and you can see that these flies are actually larger in size. Okay, so next what we did was we tried to look at why these flies are larger in size. And we really realized that these flies are actually having a delayed development. And the reason for the delayed development is because this microRNA targets one of the factors that is actually a checkpoint factor in growth and development. Okay. And by down regulating this particular microRNA, we are leading to an increase in the levels of this particular checkpoint gene, which then prevents the organism from moving to the next checkpoint. So yeah, so this is kind of where like I stop for today, but I'm going to share with you a few slides. Just give me a second. I will do so immediately, where I'm going to talk about basically my the approaches that I took and that helped me during the process of my PhD. Yeah, so share screen. Okay. Share. Okay. Okay. Yeah. This one. Yeah. So once again, I'll get you back to this work which I did in St. Xavier's College, Mumbai. Like I was saying, I was working on C. elegans, and while this was during my bachelor's, and we ended up working on these two projects during my bachelor's. And what I basically learned during the course of my this honors project was scientific rigor, because the basics of any scientific project, which is the set numbers that I would probably need. So before that, I had no clue that an experiment needed to be repeated X number of times before you actually believed it. And there was something like a power test that needs to be done to realize, okay, how many sets of a particular experiment one needs to do to actually realize whether data is significant or not, or demonstrate if the data is significant or not. And that's when actually my scientific training actually began. And we actually ended up publishing in Explore, like I was telling you before. And that's because of my wonderful professor, Professor Radhya Pacha Gupta. So, yeah. Yeah, can you see or no? This is the problem. Okay. Yeah, so that's her there. Next, we actually worked on this restriction digestion project where I was actually just doing a restriction digestion, taking out a fragment and putting it in another plasmid. Okay, so this was a project that I was working with bacteria. So this was the first time I was actually working with bacteria. And this actually introduced me to the field of molecular biology. So the previous work that I did was working on more like a systems level approach, where I was looking at the effect of various chemicals on C. elegans and its reproductive behavior and lifespan. This was my first foray into molecular biology. And oh, like I said before, I actually gave a lot of competitive exams like the TIFR, JNU exam for life sciences, JNU exam for biotechnology. And I actually cleared some of these. But when I went to these colleges, I realized that that's not for me. So this is something that you guys have to understand is we always look at the curriculum part of it and the curriculum being good, but we don't really consider the environment that we are going to be in, and that's something that you all really need to think about, because if you're not really going to be happy there, or if you can't find colleagues to actually help you move in the right direction, it's actually not going to be that useful. And obviously maintain contact with all your seniors who would be able to provide you with insight about what they have done, and would be able to give you advice as to what they have done wrong and things like that. So yeah, like I said, later on I worked in my master's in valproic acid, on the effect of valproic acid on autophagy in Dictyostelium discoidum. And once again, it was all because of my friends that I had that actually pushed me in the right direction, because uh, I was pushed. So okay, so I during my what should I say, preparation for CSIR net, okay, uh, I actually borrowed books from a lot of my classmates, because these books are really expensive, and not one can't really afford all of them. So to be honest, so I borrowed these books from a lot of my classmates, and actually instead of actually reading through them, I actually just practiced all the previous year's papers, because for me it's a process of knowing both. So you have to know matter to actually answer the questions, but you also need to realize on how to tackle these questions, and that's something that one learns when one tackles questions. So when I practiced answering all these questions, I could actually tackle questions better. I knew how to decipher the answer even if I did not know the answer. So it's more about knowing the wrong answers rather than knowing the right answers at times, right? If you know the wrong answers, the right answer becomes more apparent later on. Yeah. So that's something that my classmates and my seniors definitely helped with. And yeah, like I was saying before, and during the course of my master's here, I learned a lot of different techniques. Okay, so I learned how to do a QRT PCR. I learned how to run a polyacrylamide gel, again agarose gel. Learning was just one thing, one aspect, but I actually got to do them without supervision. So if I made any mistakes, it was all on me. And I think that is something that we need to have. We need to have more practical experience, because we really know, all of us. I knew all about all the theory behind making an agarose gel, running it, all of that, running a qPCR, everything. But I had not actually done it before. And when one actually sits to do it, and one has to troubleshoot through all the problems that one is experiencing while working on it, that's when you actually gain experience that are actually worthwhile and that people are looking for. So people they're not looking for experience like, okay, you have seen somebody do making an agarose gel, or seen somebody run a page or something like that, a polyacrylamide gel, or seen somebody do a qrt pcr. No, people are not looking for that. If you're looking for a phd, people are not looking for that. Even in the industry, people are not looking for that. People do not want somebody who's just observed things. So what I'm trying to tell you all is go out there, learn things, try to gain different experiences in different fields. And what you are actually trying to accumulate in your younger years are not projects and not papers. What you're trying to accumulate in your younger years is techniques. So that is something that I am really going to emphasize. So during my first project at St. Xavier's, I learned how to handle C. elegans and how to handle a bottle organism. Uh, Later on, and also some of the behavioral aspect, behavioral experiments. Later on, when I moved when I went for an internship to Actec, I learned about some molecular biology based techniques. Moving to Sapphires, I learned about how to read research papers. That was really important. I really learned how to read research papers, how to go through them, how to critically analyze them. And that's something that one really needs to understand. And I also got a very, very hands on practical experience in actual lab work and what it entails to actually work in a research lab. And that's something that's really important. I mean, you could get this experience anywhere, but you really need to go out there and really need to find these places that offer you such opportunities to do work, because this is actually what people are looking for. People are looking for an experience, you know. People are looking for you looking for people who have the experience to tackle problems. So like, if you run an agarose gel, and if, let's say it's not running, or you're not getting the right size band after doing a PCR, why is that? Why is that the case? And you need to troubleshoot. Okay. And they need people who can troubleshoot on their own, figure out solutions, redo the experiments, and come up with the right results. And that's something that, one needs to develop, the scientific rigor that one needs to develop. And that tends to develop when you go to a good place, which is, once again, I like to reiterate the point. The college and the environment that you do your bachelor's and your master's in actually really matter. Okay, because that's where this rigor actually comes from. It actually comes from our professors. Okay, they actually tell us what to do and how to do it, and they would actually be critical, very critical of our work. Obviously, our classmates as well are going to be critical of our work. If you're actually working, all of you are working on different projects, and then they would be critical of your work. And this actually helps improve your understanding of science, and and helps you foray into the research world as such. So yeah, so this is again something I really would like to, you know, focus on, which is, what did I do after my masters? Like I said, I gave various competitive exams like the CSIR net and the TIFR exam, and I cleared both. And I gave about 5 different interviews at 5 different places, which I actually went via TIFR, the TIFR interview process. But I finally registered using my CSIR net, because I was offered a contingency via the CSIR net, which I could use to purchase things for the lab, as well as purchase things for myself, like a hard disk to store my data and things like that. Now, this is something I really think people would be interested in, is that what do these institutes actually expect during the interview processes? And this was something that I really, really did not think about, because uh, unfortunately, all the people I spoke to before I went for an interview actually gave me their experiences, which I actually took to heart, unfortunately. And their experiences were very different from the experiences that I had. So I went to my first interview expecting to only talk about my research project during my master's, which was to look at the role of VPA on autophagy in Dictyostelium discoidum. But during my first interview, I was really thrown a curveball, and the first question that I was asked was differential equations and mathematics. So that's something that I really, really did not expect, especially in a biology interview. And I was really, really underprepared for such an interview process. And the thing with interviews, what I realized is, you should always try to get the first question right. If you do get the first question right, one actually, you know, um, becomes a little bit more reassured, a little bit more confident to address the questions that come later on, however difficult they may be. But as the interviewers, they will actually try to throw you off your game and, like, unsettle you in any way possible, so that they can actually ask different easier questions later on, and you know and see if you're able to work under pressure or really perform under pressure. And that was my first research experience, my interview experience, sorry. My second interview process, I actually studied up on mathematics, like the foolish person I am. And when I went into the interview round, I was not asked even a single question on mathematics. And the questions I was asked on were questions about a centrifuge and the max speed of a centrifuge and the difference between the RPM and RCF factor. So that was really something very different from what I experienced in my first interview session. My third interview, I was prepared. Like, I thought I was prepared. Like, I have really understood what they actually want. I think they want to know about practical knowledge and maybe mathematics and some integrative subjects. And I went to the third interview, my third interview, and when I went there, um, I was asked very, very, very theoretical questions. So the first theoretical question I was asked was, can you name the RNA pol that actually leads to transcription of various mRNAs, and which RNA pol does what? And that was really something I was not expecting, because I'm not somebody who has a really good memory, so I did not remember that. So I could not answer a lot of the questions from during that interview session. My fourth interview was at ISET Trivandrum, where I was asked much more application-based questions. So they gave me scenarios, and they asked me to apply my brain and figure out what I would do in a particular scenario. And if they gave me a question to be asked, what are the experiments that I would do to address that particular question, or perform to address that particular question? And really, I would not be able to answer these questions if I did not know about the various techniques that are available or that are present in the field. And these I learned through my research experiences at St. Xavier's, at Actrec, at Sophia. I learned all these different techniques, not only from the ones that I was working on in myself, but from all the different techniques that my lab mates were mastering at that place. Okay, so that is something that one should not take these experiences as something that you need to work towards a goal. The goal is to learn different things. Where they might actually come in use, or where they might actually come in handy, is something you cannot even fathom. Something that you have learned probably 10 years ago might come in handy later on, in later parts of your life, and you will not even know it. Okay. So one more thing I would like to tell you all about is you should read about the vacancies at a particular institute before going for the interviews. So that's something you need to know, because say if you're going to an institute for an interview, they would have a list of faculties who are actually willing to take on PhD students. And that's something you really, really need to look at, because if you're not interested in any of those professors, you really, really need to either not attend that interview, or you can attend that interview as an experience. You also need to read about what actually the professor is working on. You can't just just look at the brief gist of what the professor is working on his webpage and decide, okay, this is what I'm going to do for the rest of my life. That's something I think one has to go through. And I'm also going to tell you all this. Please ask about the professors before joining the lab. So like before joining Jishi's lab over here, I actually visited the lab. I asked a few of the students in his lab about his lab and about him. And I also asked about him, I asked various other students around from neighboring labs about how he is and as a person, as a researcher, and all sorts of questions like that. And that's something that I did before actually agreeing to join this place as a PhD student. And that's something one must always do, because during my during the course of my PhD, I have trained various master students. And one of the major problems with master students who go anywhere for their PhDs is that they never really inquired about how the PI is, or how is the, how is his temperament, how is the research caliber. So one would just look at the papers and decide, okay, this guy is publishing well, so I probably would join him. But like I said, the environment of the place really matters, because it would stimulate you to do good science, better science in life. Yeah. So once, last but not the least, I would like to tell you that during the course of my PhD, I had to do a lot of QRT-PCRs uh, for my work, which was actually something I actually learned during my master's at Sophia, and actually helped me, because now I can use, so we had a different instrument at Sophia. We had an ABI instrument, and here we have a Biorad instrument. But I'm experienced at using different qPCR machines. It's not actually a difficult task. And that's because I have got the practical experience of using these machines. And like I was telling you before, during the course of my PhD, we had to shift our labs from one um, place to the other. And during the course of my PhD, a lot of our instruments broke down, and I had to continuously change my projects. But what I'm trying to tell you all here is, look at the silver lining. The silver lining is one has to always be adaptable. And the current state of the world is, if you don't have money, you cannot do research. And that is absolutely untrue. Uh, in terms of research, you would have to address smaller research questions. Yes, definitely. The impact would be lower, yes, but you can definitely make a contribution to science. And one must always be flexible when one is choosing one research idea, because like I said, a PhD is just a training process. And in the end, I would like to, uh, end by saying that this is what I did from 2015 onwards. I presented my poster at various research conferences held in India, both international and national, and that's something you should always try to do. I also catch various people who come for talks here to ICER. I catch them when they're free, and actually present my work to them and ask them for suggestions. And that's something one must always do. One must also look for avenues outside PhD to promote your own work, which is why I wrote a short story for DST officer. And one must always remember to work on one's hobbies, because this is something that one must always remember. Research work is a marathon, not a sprint. So you have to work at a constant pace rather than work at a breathtaking pace and actually burn out. And that's something that one must always do. And you should also volunteer for various research activities and various other activities, and help while hosting a conference and things like that, because this helps you make connections, because connections are really important. Okay, so that is something one must really make in life. And I'm going to end on this note, that it's so near yet so far on my way to the home plate. But I would like to add that it's still the first innings for me. The PhD that I'm going to complete is just going to be the start. And this is just a training. And later on, I would move to actually doing work of my own that would be the second innings, and further on the whole game. So thank you. And yeah, I'm open for any questions.
Thank you very much, sir. That was indeed a very interesting journey that you shared with us.
Yeah.
And yes, we will now proceed on to the question answer session.
Mm-hmm.
Over to you, Lenisha.
Thank you, sir, for the exceptionally informative session. I hope you all enjoyed this amazing talk. Now we can begin with the question answer session. So, sir, our first question is, could there be a functional role of the studied microRNAs in other organisms in gene editing?
In gene editing, not in gene editing, but in gene regulation, yes. So like the microRNAs that we are looking at, So like the microRNA I'm looking at, they are conserved across bilaterians, which means they're present in humans as well. So this microRNA, this particular microRNA that I'm looking at, it actually affects lifespan only in male flies, in flies. And it's known that the gene that's actually targeting, these genes are actually conserved in humans. Okay, that's something that I did not talk about. And that's something that mammalian guys would look into, whether these particular genes actually play a role in lifespan or not. That's something for them to look into. But what I'm trying to say is that these are actually having roles to play in humans. The last project that I was looking at was the role of microRNAs in growth. And this particular microRNA actually targets, like I said, it targets one of these proteins which regulates a checkpoint. And these checkpoints are actually involved in development, but it also plays a role in various epithelial tumors, which is what this tissue is actually, where it's expressed in actually is a homologous form. And this microRNA actually could be playing a role in regulating its ortholog in humans, regulate in epithelial tumors. And that's something, once again, the mammalian people would look into. Yeah, so that's not part of our work. So we are doing basic science. So we actually pinpoint factors that actually play a role in Drosophila and a lot of different, and could play a role in humans. But it's up to the mammalian guys to look into whether these actually factors actually have a role to play in humans or not. I hope I've
Thank you,
answered your
sir.
question. Yeah.
Thank you, sir. Our next question is, how did you manage to look for another approach when you decide to do one particular project but have to shift to another? What is your strategy to look for another project of your choice and likings?
Uh, yeah, the thing is, one has to understand is that uh, like I said, you should always look to learning new techniques, and it's all about techniques rather than the project and the techniques that you learn during your PhD. Because to be honest, to be really honest, any paper that you publish in your PhD is not it's not attributed to your genius or your brilliance. It's attributed to a contribution of yours as well as the PI's input. Whereas later on in your life, whatever you do after your PhD, during your postdoc or other research experience, is attributed to your, your scientific rigor and your, interests and things like that. So most of the times, what you're working on, you might start your journey in one particular direction, but you might end up in something completely different. And that's something that's observed when you have longer PhD programs, like a 5-year PhD program. You can end up in places like this. When you go to Europe or places where they have a 3-year PhD program, it's a much more structured program. So you end up with working on exactly the thing that you're looking at. And that really doesn't actually, for me, it wouldn't be a good experience, because one should always try to understand different things and try to get different experiences. And one of the major things that I've learned on is not how to pick good projects, it's learn how to figure out which are the bad projects and how to drop them. So I've only spoken about 3 projects, that these are the 3 that worked. That doesn't mean that I have not tried a lot of other projects which I haven't worked, which haven't worked at all. And yeah, so what one should try to understand more is when to drop a project, what are bad projects. Those are a lot more important in terms of all of this. And good projects, I think one will get interested in work when they're getting good positive results. Automatically one is inclined towards working in that particular project, is what I feel as a person. Yeah.
Thank you, sir. Our next question is, how is it like working in the COVID facility?
Oh, COVID facility. It gets hot sometimes because of all the PP. That's all. Because the experience is actually the same as working in a lab, because it's just a QRT-PCR. So RNA extraction and QRT-PCR, which is exactly the same what I do in the lab. But it's just that I have to wear a PPE suit and work in a different laboratory, which is not connected to our main campus. That's it.
Thank you, sir. Our next question is, do you recommend that we take a break before starting PhD to gain experience?
Recommend take a break. It depends on person to person. Like I said, once again, PhD is just the process, and one should really, really learn, techniques. And okay, I'm going to be once more. I'm really going to be honest with you. I don't think one really needs it thinks about the fact that what will one do after a PhD. And let me tell you, a lot of jobs out there have age criteria in India. So they have an age cutoff of say 35 years old. So if I go to ISC after 35, they will not even invite me to actually give a talk there and try to join there as a faculty. So I cannot even give an interview there to join as a faculty at after 35, and that's something that's there. So one must keep that in mind, because that will in the long run affect the entire process. But definitely don't rush into a PhD. Like I said, follow the steps, ask around. If you get this interview experience is really important, because like I said, I really did not expect any of these questions. Okay. And like my first interview was completely different from my second interview, from my third interview, my fourth interview. And now I have a lot of students who are actually master students who apply for PhDs abroad, and their experiences are really different. I've had uh, students who have been asked questions like whether they'll what's their personality like, and that was the only question that they were asked, and whether they would be able to gel in the lab. Those were the only two questions that they were asked. So these are really really different experiences, and one needs to really go through it to actually understand how to tackle interviewers and answer their questions properly.
Yeah, thank you, sir. Our next question is how to apply for project assistant. Are there any exams for this post?
No, actually, those are going to be just vacancies given on the institute websites. So all the individual institutes. Plus, Biotechnica is a good website that actually sends you emails regarding all the job positions and all of these things that are available in the field. So I think one should definitely subscribe to that to keep keep this in the background, and you can always apply to all these places whenever you want. Yeah.
Thank you, sir, for answering all our questions. This being our last question,
Yeah.
now I would hand over to Chandrima to propose a word of thanks.
Gratitude is the blossom that springs from soul. A very good evening to one and all present here. On behalf of Department of Life Sciences of Sophia College, I would like to express my gratitude towards our honorable speaker, Mr. Jervis Fernandez, for spending his valuable time with us and giving his insights. Starting your projects from a very young age, as young as just after completing your 10th standard, was extremely encouraging for all the young scientists out there. Then your journey from Xavier's College to Sophia and then ISR Trivandrum was commendable. You faced so many challenges during your PhD and you had to change your project so many times, but you did not lose your motivation and hope, which is very inspiring. um, and you working as a COVID tester in ISER Trivander makes you a COVID warrior, which is very applaudable, and our gratitude towards you. Your experiences are lessons for us. The talk was very enlightening and very helpful for our students with full of knowledge and interesting facts. And finally, thank you so much for patiently listening to our questions and answering them properly. Participants kindly copy the feedback form posted in the YouTube chat box. It will be activated after the session for 30 minutes. Now I request Dr. Hema Subramaniam ma'am to invite our next guest.
Well, hello everyone. And from one beautiful lecture talk to another, we warmly welcome Priyanka Sigar, who has been with us as an MSc student. We're very happy to have you with us, Priyanka. She has been a very sincere, plodding student who went through her master's very excellently, and we're very happy that you are able to join us today. I must say that Priyanka has got a very talented artistic side to her also that she has displayed with us. So I hope that's also progressing well, Priyanka. So do proceed. Now I will hand this over to Astha for introducing you formally, and then you may please start your talk. Asta, over to you.
Thank you, ma'am. I would now like to take this opportunity to introduce our first speaker for the day, Miss Priyanka Sigar. Miss Priyanka Sigar completed her master's in bioscience in the year 2008. She is currently the project associate on a project entitled Temporal Stability of Functional Brain Networks Associated with Autistic Spectrum Disorder at NBRC, India. She has previously done several internships from various renowned institutes like TIFR and the Homi Bhabha Center for Science Education. She also worked as an intern at Little Pods, an organization for special kids in India from January to June 2019. She has given a talk on urbanization and its effects on the environment and the role of nature in restoring mental health for Northeast India Conservancy. She has volunteered for many different campaigns and workshops. She is also currently conducting workshops on career in research and data analysis methodologies. She is also a student member of Indian Science Congress. We are very happy to have you with us, Ms. Priyanka Sigar. Before we move on, I would once again like to request our participants to kindly type in any questions that they have in the YouTube chat box, which will be addressed after the talk. I would now request Ms. Sigar to take over from here.
Thank you so much for this introduction. It was pretty overwhelming, and especially thanks to ma'am because you remembered my music side. I connected with you on those drums basically, and I'm really thankful for this opportunity to be able to give back something to my alma mater because Sophia was something that changed my life a lot in terms of my career. And yeah, I'll tell you about how, my career goes switch from one domain to another, and then where can you possibly land after your master's. So before starting with that, I would like to know if I'm audible. If in case my internet is working properly, I hope my voice is not cracking. So can anyone give me the feedback for that?
Yes, ma'am, we can hear you.
Okay, that's great. And now I need to figure out how do I share my screen. Okay. Is my screen visible?
Yes, ma'am, it is.
Okay. So starting with, as mentioned, I'm a project associate at NBRC right now, and I completed my bachelor's from CHM College in zoology, and then shifted to master's in neuroscience from Sophia College. For that, motivation came from Dr. Pankaj Seth, who came to our college in bachelor's to give a talk on how neuroscience is conducted, and then I had a lot of my seniors who came to Sophia for getting their master's in neuroscience because it is one of the fabulous courses that I'll be talking about. How 2 years get utilized in a proper way? I'll try to emphasize on that a lot, which most of the institutes miss out on. I'm telling you, you're in one of the best places if you really want to pursue a career in neuroscience or be in any other domain. In terms of research. After my master's, I did my internship at this institute wherein I was working with the CRISPR-Cas system because it was pretty new at that time. But it was my master's in neuroscience basically at Sophia, which motivated me to change my domain to, studying kids with special needs like autistic and ADHD. And that motivated me further to work with autistic kids and their, the brain activation domain. I'll just try to change this. Okay. So currently I'm working here since 2 years, and now I've applied for PhD, and I'm hoping to be in one of these places for my PhD in September or October. So yeah, starting with my journey. Sophia has been my milestone in terms of my career. So how 2 years were constructed, it was really rigorous. You will feel that, you're under a lot of stress and a lot of pressure in terms of work. But I'm telling you, it's one of the well-structured courses that you'll experience in terms, not just theory-wise, but in terms of your practicals, in terms of your research, in terms of your writing skills and research that you require, which you'll gain via your literature review and writing thesis. So basically, your 2 years, you start getting training from your 6 months or first 6 months of your masters, and then you continue to do that for like the rest 2 of your years. And then you do a lot of literature review work along with your research, and then theory and practicals, are rigorous. And the diversity of the curriculum that you have, ranging from be it biological neuroscience, computational neuroscience, cognitive. It has a lot of variety. For you to choose what you want to do in your PhD ahead, it just ranges from selecting an industry to going to research domain. It gives you a lot of scope what you want to do after your master's. And it is pretty collaborative. You must have gone to TFR for a lot of conferences for that matter. So yeah, this was my master's journey and my master's batch, wherein a lot of us did a lot of work in different domains. We had Xenia working on chicken Geo, then we had Cheryl working on Hydra. I'm sorry, Cheryl, for the exclusive photos. Then we had Dhanishri and Anjana working on Dicto, and Ikra and I used to work on zebrafish. I also used to work on cell culture. We had Neha working with C. elegans. Shweta with Drosso, and we had a behavioral person. So this is our pre and post photos. This was like our during master's photo, and this was our graduation day photo. I'm really thankful for help, especially to our faculties, and then we had Nabila ma'am and others, Mayuri, helping us with most of our projects. So talking about my project focused on effect of lithium acid drug on the migratory behavior of glioma cells using zebrafish xenotransplant models. And I worked under Dr. Yasmin Khan. So wherein first what I did was I wanted to study how the morphology and the proliferation of these cells can be controlled using lithium. So this is a control photo, and this is when it was given treatment with lithium. Then we wanted to study if in case we do see these kind of changes, what is actually leading to these changes. So we wanted to study how your actins are organized in these cells, which leads to this kind of differential morphology when lithium treatment is given. So in case of controls, you'll see that actin is organized randomly in the cells everywhere. But in case of post-treatment, you will see that it is pretty well organized, giving the cells back their morphological shape that was desired. Then what we did was, so my seniors also checked how these migrate differently. So we wanted to check when these cells are injected into zebrafish, how would they migrate in a given system, living system, and how they would proliferate. So what we did was we micro-injected these cells, which were dyed red using dye, and these were injected into these larvas. So these are the results for that. Yeah. So this was post 2 days of injection, wherein this is the yolk area that you see, and in the tail area that you see, the cells have migrated. After 5 days of injection, the cells have proliferated even further, and you'll see same amount, increased amount of cells in the tail region though. So basically, I worked with 2 model systems. I had to take care of the cells, I had to take care of the zebrafish, and yeah, it was a lot of work, but then it seems pretty fancy and serves a lot of purpose in terms of being able to contribute to the society if we can provide any drug for people suffering with glioblastoma cancers. So this was our journey. We got to do a lot during our masters. We attended a lot of conferences. In my case, I was restricted to one because of other issues. This is a photo from ACTRACT where we went to present our work. This was our talented group where we had a lot of dance performances from our group, and yeah, me and a couple of friends were singers, so we tried to show off our talent there. And then this is our cultural day photo. Yeah. There was a turning point which happened during masters. So in masters, you have a volunteer experience wherein you go to Sadhana school and you work with special kids. You get that experience, I guess, for 1 month. So that's what we got to do. We went to Sadhana school for that kind of volunteership. I worked with a kid with Down syndrome, and along with that, we were provided course on how neuroimaging and other imaging techniques can be used to study these disorders. And we also had a bit of course on psychology. So that really interested me in a way. How can I contribute to the society by working with these kids? Because I really enjoyed working with them. Just 1 month of experience really, gave me a thought that whether should I change my domain, stay with the cells and work with in the wet lab, or should change my domain entirely. So I changed my domain, and this in my CV really worked out. That's the reason why I got my another job for 6 months as an intern at Little Pots, wherein I worked as an intern as a child counselor, or call it a shadow teacher, or maybe a therapist, wherein I was trained under a senior clinical psychologist. And I was trained how to give these kids behavioral training, how to help them develop academic skills. So you are constantly under training when you're doing these kind of jobs. You have to be with these kids. So I was allotted a kid with developmental issue. He had autism also. He was a 4 years kid. So I had to teach him how to behave in a social scenario, and I had to monitor his behaviors. So I used to spend 7 hours of my day with that kid, basically noting his behavior, providing him therapies, and reporting to his parents and my seniors about the development basically. After that, being a neuro researcher, I was really interested in understanding when you give these kind of therapies, what actually changes in the brain in terms of your brain activities, activations, how the brain regions are connected, how that changes in these kids when these therapies are provided. So that's the reason why I came to National Brain Research Center, wherein I'm working at the Cognitive Brain Dynamics Lab under Dr. Deepanjan. So wherein we look how in autistic kids, basically these functional brain networks are organized in these kids. So here what we do is we do a lot of projects in the lab, wherein the major focus goes on sensory processing and perception. There's aging and development, cognitive flexibility in atypical individuals with autism. And then we do a lot of mathematical modeling and how you create a simulated model and then give it a stimulus, and then you see what kind of changes occur basically. If in case you have data, you want to validate it, you probably create a model, and then that's how you validate your data driven methods. And then we do a lot of behavioral experiments wherein we use EEG to do those kind of reports. We have eye tracker also, and we have fMRI studies going on. So talking about my own project, I would like you to have a brief understanding of what these brain networks, how these brain networks are organized. And here node means brain regions. So from nodes to brain regions. So for example, your brain regions are connected differently and functionally connected differently in a given task. And these come together to form different functional networks. These networks then form group again. They integrate and segregate from each other to form modular structures. So if in case there is a particular task going on, your brain actually needs to integrate and segregate differently so that it can carry out that task. In these modules itself, there has to be efficient communication within the modules and across these modules. And what takes care of these kind of communication? Your different nodes. There are few nodes or few brain regions who will take care of the communication which is happening within the networks, within those modular structures. And there are few which will take care of the communication between and within these networks. So my main focus has been to understand how the local brain areas can affect these organization of functional networks. So why specifically autism in that case? Because autism is a disorder of several networks. The word autism itself means being locked within self or being removed from social interaction. It is characterized by impaired social communication and repetitive behavior. So these are the two major traits. But why do we call it spectrum? Because there can be a variable degree of dysfunction in these two categories of impairment. So I would like to give a basic example so that you have idea about functional networks and how those can affect your social communication behavior. So these are three examples of functional networks, like your DMN, that is majorly in your frontal area, which takes care of processing and inner mentalizing processes like how would you think about yourself, self-referential, autobiographical thinking about yourself. Your salience network will take care of switching from one task to another. Then your control executive network will take care of integration of information that comes from different networks basically. So here we'll try to understand how social communication can be affected just considering these networks. For example, there is some social input that comes and salience is like this is one relevant stimuli that needs to go to the DMN for processing. So it is processed and that information is integrated by your CEN network. And then further, whatever the output is there will be executed. And if in case there is any issue at any of these network level or at the communication level of these networks will lead to social communication deficit. So talking about my project, I study how alteration in these brain network organization actually arises from alteration in the role of local brain areas in autistic kids. So this project is, I'm primarily working with Dr. Deepanjan and this project is in collaboration with Dr. Lucina from University of Miami. So wherein what I do is I don't collect the data by myself. There are a lot of open source databases like Abide here, wherein people collect data from different subjects and then they upload it on these websites basically, and anyone can use it to do different kind of studies. So I use Abide to carry out my study. It is a resting state data wherein you don't have to do any tasks. So basically during the resting state, it is known that even when you're not doing different tasks, your brain actually keeps on working. Majorly your DMN, wherein when you're quiet, you're not doing anything, you're thinking about yourself. So I'm using the resting state fMRI data. And for data pre-processing, like your fMRI data could be a raw data wherein you have a lot of head motions and you have a lot of noise. So you need to remove all of those things. So data pre-processing is required. And after that, I do a lot of coding using MATLAB, which I use for data analysis. So for my project, what I've done so far is first I've looked at the global studies wherein I see what brain regions are connected to each other and then how these brain regions come together to form different modular structures and then how the brain areas could contribute to these kind of modular organization. So this is my lab wherein we go to a lot of trips so this is my lab to sum it up. And yeah, talking about the scientific research, you think that you're going to solve a problem basically. Then you think you have solved the problem. But then when you analyze it, you actually realize that the problem is further more complex and then you need to answer it. And for that, going back to the loop one. So yeah, what can you do after a master's? You can get into science communication. You can write about science. You can draw. So I have Insta page. I get offers from different articles to draw for them. So I do graphic designing also for them. I draw a lot of logos. So you can do that. Then there are different industries. So like I do coding a lot. Since you're in neuroscience, you have a neuro background. So like companies like Facebook and Google, which need to understand how people interact with the different websites, how they look at different products. They need to understand that, and then they need people from neuroscience like us Then you can get into neuromarketing. So a lot of friends of mine are there into neuromarketing, wherein you look at a product and then how do you think about it? What would compel you to buy it? So there are a lot of studies going on. So wherein you use eye trackers, what things of the product do you like a lot? What do you stare a lot at? And you do EEG along with that. So neuromarketing also needs a lot of neuro researchers from our domain. Then you can move to psychology. Maybe like me, you can directly join as a shadow teacher or as a child counselor, basically intern there. And then you get promoted to at different stages, and you can be a permanent if you really like that job. If in case you want to move to psychotherapy, so basically you can get a diploma degree in that, a 1 or 2 years degree, and then you can, I guess, be occupational therapist. So there are different kind of courses. If you want to get a master's in psychology, then either you can go abroad and pursue one because they give you training in that, um, or you can do a PhD after your master's, wherein they give you a license by helping you to develop those counseling skills. Or you have a lot of distance courses also, like Indira Gandhi University provides you a distance course in master's, so you get a degree from that. Or you can get into neuropsychology, like what I'm doing right now. And if you want to do a PhD in this, know what you want to get committed to. Like after my master's, I switched fields. I went to fundamental foundation of medical research center to see if I really want to continue in this domain. Then I switched to being a intern at Little Pots to know what I really like. And now I'm here since 2 years, and now I know that I'm prepped up to be in that commitment of or maybe I'm ready to be committed to PhD. So I know that I am now ready for PhD. So I've applied for it and I've got offers from 2 places. And then when you know that you are ready, you approach a PI. Basically, you read about their work, you write an email to them, tell them what you like and possibly what kind of work you want to do, and then tell them that what kind of scholarships you can try for. Like UK needs you to have certain scholarships by them. UK universities actually want you to be prepared with what certain kind of scholarships, but in USA they provide you funding, so you don't have to look for any scholarships. Like UK, Canada situation is there, and in Germany. So basically, depends upon what place you are applying for, you will have to look for different scholarships, maybe. Yeah, so that's it about me. Yeah, I'll stop sharing my screen, and yeah. Hello?
Thank you. Thank you very much, ma'am. That was a very informative session and a very inspiring journey. We will now proceed with the question answer session. So over to Anisha.
Thank you, ma'am, for the informative session. We are grateful to you for the time and effort you took to share your knowledge with us. Now we shall proceed with the question answer session. So, ma'am, the first question is, what made you choose glial cells as a topic under the model organism zebrafish during your masters?
So basically, during my master's, what happened was we already had PIs working around certain projects, and we were supposed to have that experience of how research is conducted. So basically, I did not opt for it; it was already there. I just went in because it looked really interesting to me in terms of cell culturing and the glioma. It's your brain cancer disease, basically. So I had an option to select it, and so I went ahead with it. Yeah.
Thank you, ma'am. So, the next question is, what has been the effect of early intervention and therapy on children with ADHD? Also, could you highlight the same with respect to fMRI studies?
Okay, so in kids, it's known that right from 3 months of stage, you can detect ASD. So if in case treatments or interventions are given from that early stage, they can start to develop those better social communication skills, and you will be able to help them assign tasks to prevent repetitive behavior. So both from fMRI perspective and from the behavioral perspective, If in case the parents are able to diagnose ASD at the early stage, and if they start interventions at home, the chances of helping these kids cope up is far better.
Thank you, ma'am. Our next question is, could you highlight the relation of plasticity of neurons with respect to early diagnosis in ASD?
Okay, in kids, actually, neuroplasticity is pretty faster because if you people have done a lot of study wherein they have looked at kids, teenagers, and adults. So a lot of neuroplasticity happens when the kids are developing from childhood to say teenage. So in that case, if in case interventions or diagnosis are availed at that stage, basically, so that really helps that progression from childhood to teenage in terms of neuroplasticity.
Thank you, ma'am. Our next question is, which internship courses would you recommend to our aspiring postgraduates?
Internship courses in regards, I guess it's not really that specific. See, depends upon what you want to explore after your master's. Since it has provided you a diverse curriculum, you know what all exists. So basically, you choose what you like, and then you approach certain PIs, and you can write to different PIs in different institutes in India or maybe in abroad also. They take you for RA position and PA position, and then you can continue for your internship basically like that.
Yeah, thank you, ma'am. Our next question is, could you tell us about any scholarships or grants after masters that you would recommend applying?
See, one few that I'm aware of are the Erasmus Mundus that you will need for European or the Germany institutes. If you want to go to UK, basically you'll need Felix scholarship. Then there are a lot of scholarships that are provided from India, like the Tata scholarship is there, and the Young Women Scientist scholarship is also there. If in case you progress well and you have a lot of good academic scores, basically. Then you have Inspire Scholarship Fellowship after your master's, actually. So it depends upon where you want to apply. Depending upon that, India also has a lot of scholarships and fellowships. And if in case you want to go abroad, there are a couple of scholarships wherein Indian government collaborates with these universities. So if in case you want to go to Columbia University, that university also is in collaboration with the Indian government. So it provides money to kids in India who want to pursue their PhD in Columbia. So yeah.
Thank you, ma'am. Our next question is, in what way does placement or internships help our future goals?
Um, Placements. Okay. So in my case, know what you want to do in your future. Like I want to get into startup basically. So I know what I want to do. So for that, I got into internship as a child counselor, and now I'm working as a PA here to get my CV stronger so that when I reach that stage, I will be able to say that, take me because I have these skills appropriate for this job. During my PhD also, I will be also trained as a counselor. I will be able to work for industries. I'll have teaching experience. I'll have research experience. So depending upon what you want to do, you need to list out what skills you need to develop. Get into those jobs, get those skills, and then that's it.
Thank you, ma'am. Our next question is, what is your perspective of making use of your hobbies to pursue your career and keep it as your co-curricular option and find it helpful and related to the career field of your choice?
That's a really great question. So basically, I did not know if I could use my art. So I was into drawing previously and I did not know how to use it. But then when I got here, I have a couple of friends who write for articles and magazines. So they need a lot of logo work basically. So depending upon what your skill is, you can also get in, if in case you're a musician, you can do a lot of research based on music therapy or how music, different ragas can actually bring about changes in your functional brain network. So what your skill is, you have everything that you can relate with in neuroscience. If you want to do research related to your hobby, you can do that. There are a lot of people who study that when an artist looks at a particular drawing, how his brain perceives it differently, and how person who does not really understand drawing or who does not really like drawings, how his mind will perceive it differently. So one way to be with your hobby is to do research in it, or maybe find industries which actually pick up your hobbies and collaborate it with science communication. Yeah.
Thank you, ma'am. Our next question is, do we need an education agent or a student counselor to guide us through the process of choosing the right university?
That is a full-time scam that is going out in the market. You will have a lot of companies which will tell you, we can get you into certain universities, and these are the ones you should apply to. And these are the GRE and the TOEFL scores that you need to get into these universities. That's a total scam that's going on. I'll tell you, I did not have good GRE scores. I had decent amount of TOEFL, yet I managed to get into this university majorly because I knew my PIs. I was in contact with them since a year. They knew my work, and since these companies actually do not know what you want to work in, they just suggest you average institutes which will take just any other student, so your guarantee of getting into this universities increase. But then when you're doing a PhD, you actually need to know what you want to commit to for those 5 6 years. So it's your job basically, and you should not really rely on those people, because they just feed higher expectations in your minds. If in case you want to get into places like Miami, you need to have GRE scores reaching heights, and TOEFL, needs to look pretty glittering. But that's not the fact. During a PhD, your PI matters, your research interest matters, and how you have developed your research skills. I'll tell you, there are a lot of people who did not actually also have a lot of research experience in the field that they wanted to get PhD in, but they knew what they want to do. They did a lot of research, reading of those PIs, and they saw that these kids are, excited and motivated enough to pursue a PhD. So during the PhD, it's a learning process anyway. So you'll get those skills during a PhD. So don't go to these companies. I'll tell you, they'll just, ask you for money and guarantee your admissions in a place that you don't really deserve. You deserve to be in a really good place, actually. So, yeah.
Yeah, thank you, ma'am. Our next question is, did you take any courses in neuropsychology?
No, I'll tell you, Coursera is a place that if in case you're motivated and if in case you don't have money, you can tell them that I don't have that kind of money. You write them whatever email, they'll give you financial aid, they'll give you a certificate for the courses that you've applied. So I did all my MATLAB learning from Coursera and from YouTube, and most of the pre-processing understanding and the neuropsychology understanding that I required from Coursera and YouTube itself. So, see, if in case you go for an interview, they'll understand what you understand, what you know actually, and how motivated you are to, to apply your information and to learn more from it. So you don't really need to go for those highly expensive courses. But if in case you want to, then you can go for 6 months courses. Different psychology centers actually provide you those courses, but those were too expensive for me. And I happened to just, use Coursera and get to the places that I wanted to go to.
Thank you, ma'am. Our next question is, have you worked with other psychological disorders like AHD?
Yeah. I've worked with autistic kids, I've worked with kids with cerebral palsy, then with Down syndrome and ADHD.
Thank you, ma'am, for answering all our questions. With that being our last question for the day, I would like to invite Ruchi to propose a word of thanks.
Good evening, everyone. I take this opportunity on behalf of the Department of Life Sciences, Sophia College, to extend my gratitude to Ms. Priyanka Sigar for having accepted our invitation at very last moment and joining here us today. Ma'am, we are very grateful and enlightened listening to your research experiences in the field of life sciences. Thank you, ma'am, for sharing your images and explaining your zebrafish and glioma cell culture work so explicitly back at Sophia College and your brain's network organization at NBRC. It is really
Thank you.
motivating to see how you work. Yeah, yeah, ma'am. It is really motivating to see how you work with special kids and giving back to the society. It makes us aware about our social responsibilities as well. Thank you, ma'am, for shedding light on autism spectrum disorder and giving us some insights in your work. You also helped us get a clear picture about options available after MSc, which is often a question mark to many students. This being said, I once again thank Ms. Priyanka for answering the questions put forward by our participants and leaving us inspired. Thank you, ma'am.
Thank you so much for inviting in the first place. Yeah, it means a lot. And to be able to give back to the place that has, motivated me to change my career and be where I am. Yeah, I'm really grateful for the opportunity. Thank you.
Thank you, ma'am. Now, proceeding further, we thank our college and our principal, Sister Ananda, for giving us this opportunity. We also express our gratitude to our vice principal, Dr. Yasmin Khan, and the head of the Department of Life Sciences, Dr. Hemlata Ramchandran. Without their constant support and encouragement, this event was not possible. Today's webinar was supported by the DBT Star status, for which we are very grateful. Thank you to all our participants for joining us for this 2-day webinar and making it a collaborative learning experience. We hope to see you in future as well for the upcoming webinars organized by our department and benefit from it. Despite this being an online session, organizing such events take time and combined efforts of many individuals. On behalf of MSc Part 1s, I thank our professors and other students for their insights and help in all possible ways to make this event a success. Lastly, viewers, kindly note that the link for feedback form has been put up in the chat box and will be active for next 15 minutes only. Make sure to fill up the form in order to receive the e-certificates, which will be mailed to you on your registered email IDs within 1 week. Thank you.